people who are on the course at Manningley. Good to see you. My name is Bob White. I'm director of the Faraday Institute, and this lecture is one which we have every term, but it's linked to the course this time. And today is a very auspicious day for electron biology. As you may not know, it's Darwin's 207th birthday, and I think he's bearing us pretty well. So that's today, but today we're going to have Jeff Hardin talking to us. He's Professor and Chair of the Department of Zoology at Wisconsin-Madison in the States. He's come across from there. And he's very well qualified to give this talk tonight, which is a joint Faraday Institute and Christians in Science lecture, because he has a Master of Divinity from the International School of Theology, and he has a PhD in biophysics from Berkeley. So he's embraced those two hearts, and we look forward to hearing what he has to say on this issue of biology and belief. But just to tell you a little bit about what he does, his research focus is on the genetic regulation of cell movement and cell adhesion as embryos develop. So that obviously has a lot of implications for understanding birth defects in humans and also cancer. And he also runs an organisation called the Isthmus Society at his university. He's the only scientist apparently in the religious studies program there. So he really does embrace these two fields, which is the hallmark of what the Faraday Institute does. So we're very pleased with that. He also is on the board of Biologos, which is an American organisation which looks at many of these issues between Christianity and biological things like evolution. I think he left a few leaflets like this that are on the table at the back. So the plan today is that Jeff will give us a lecture for 50 minutes or so. There'll be plenty of time for questions after that. Then we'll have a reception out in the foyer there. So without further ado, Jeff, over to you.
Let's see. I'm going to get my laser pointer. Hold on. I'm armed and dangerous. Great. Well, it's a great pleasure for me to be here. What an honor it is to be here as part of this biology and belief workshop. I want to thank Ruth and Kara and the entire Faraday Institute and CIS for making this possible. Actually, I'm a member of CIS, for those of you who are in CIS, so some of us are more enlightened across the pond than and recognize what a tremendous organization that is. And it's really, I think, been a dream of mine to be over here and interact with all of the great folks at the Faraday Institute. I appreciate so much all the work that the Institute does, and only wish that we had something even remotely approaching what the Faraday Institute has been and is doing in my own home institution at University of Wisconsin. When Ruth invited me to give this public lecture, I will admit to being a little bit perplexed in that the goals here are to talk a bit about the basic science of the discipline that I represent. I'm something called a developmental biologist. I study how embryos develop. Then to say a little bit about my own research, and then to make some connections between biology and belief. And that's what I hope to do. That's the goal here. And so I hope that in the question and answer time we can have a really rich discussion about these topics. Well, I begin my spring semester developmental biology course to my upper division undergraduate students by telling them that I have one fundamental goal for the entire semester. They will likely forget much of what I discuss in terms of the molecular and cellular mechanisms of embryonic development. But if at the end of the semester they look back on what they've learned and they have the fundamental feeling that embryos are cool, then I've succeeded. And that's because for me, being a biologist is about having a sense of wonder about the living world and being able to celebrate the amazing complexities of that world. And as we'll see, as a Christian who is a biologist, that ultimately leads me to a sense of awe and wonder that really is worship. But for my students, I want them to have a sense of wonder, and if you're a parent, as I am, you can't help but be amazed at the regularity of developmental processes. This is my son Christopher, 27 years ago. He had a birthday this week. You know, when you look at the amazing structures that are formed as a result of embryonic development, you can't help but be amazed. The regularity of formation of things like the tiny digits that Christopher has in this picture, the structures from head to toe that a baby represents. It's that reproducibility and the sense of amazement that one has when one becomes a parent for the first time. It's the kind of thing that I want to inculcate in my students, and I hope you get a sense for the amazement that developmental biology really is as a result of tonight's lecture. Well, people have been thinking about this for a long time. Let's just take one example. This is from the Hebrew Scriptures, some poetry ascribed to a fellow named David, and it goes like this. For you formed my inward parts, you wove me in my mother's womb. I will give thanks to you, for I am fearfully and wonderfully made. Wonderful are your works, and my soul knows it very well. My frame was not hidden from you when I was made in secret and skillfully wrought in the depths of the earth. Probably a metaphor there. Your eyes have seen my unformed substance. J.R.R. Tolkien knew a bit of Hebrew. The word there is golem. Gollum from the Lord of the Rings gets his name from that word. And in your book were all written the days that were ordained for me, when as yet there was not one of them. So notice that this Hebrew poet understands some fundamental things about embryonic development. First, it's a process. You wove me, that word has to do with being knitted together. There's something processive about that with them. And it involves construction, just as knitting a jumper does for us today. And he's also amazed at it. Now, he didn't understand anything about early development, and that he has a sense of the awesomeness, the amazing qualities of embryonic development. And that it begins with something that's not fully formed and leads to something that is fully formed. Now, for him, he has no ideas about the mechanisms that lie behind embryonic development. That's the tremendous privilege that we have living in the 21st century. When you think about it, building an embryo is a staggering challenge. We all started as a one-celled zygote up here on the upper left, and some complicated things happen, and then you get something really complicated at the end of this. You know, a human infant has hundreds of specialized cell types, and it has about 5 trillion or so cells. A full-grown adult has on the order of 50 trillion cells. So think about that for a moment. And you recognize the challenge of early embryonic development. Now, it turns out that much of the key work that's done to set up the basic body plan and the differentiated structures of an embryo happens during very early development. And that's what I'll focus on tonight. Now, there were people thinking about this process for a very long time. And let's just do an experiment here on stage. We're going to do an experiment using the tools available to a fellow named Aristotle. You may have heard of him. Aristotle was tutor to Alexander the Great. And he used a fundamental tool, bird's eggs. And he cracked them open. And he looked. Now, when you look at a bird's egg, well, besides it being a mess in your hand, you notice, you can see that as birds develop over time and you crack open more progressively fully formed bird embryos, that initially you can't really see any perceptible structures with the naked eye anyway. But by the end, you have incredible complexity of embryonic structure. And so Aristotle thought, well, that's pretty interesting. And he started thinking about how that would work. And he postulated that there had to be some sort of interactions between the component parts. The simpler parts leading to greater and greater complexity over time. And he called that epigenesis. That complexity arises through the interaction of these simpler parts. And Aristotle was really right about this fundamental process. And in fact, this idea of epigenesis ruled throughout antiquity through the Middle Ages, until the invention of the modern microscope, and people began poking around looking at cells, and they began to believe that they could see incredibly intricate structures in those cells that they were pondering, including the head of a sperm. So later you have this idea known as preformation. So this is think of this as little mini-me wrapped up in the head of a sperm here. This is called a homunculus. And so people thought that, hey, maybe, in fact, early development is all front-loaded into the sperm, and the egg is just a storage dump of nutrients or something like that. Now, we know that Aristotle was closer to the truth than the preformationists. But it is true that components are put into the egg, actually, that help to direct early development. And I won't have much to say about those because we're going to focus on bodybuilding in what we're saying today. Now, I do want to point out one thing because of what I'm going to say through much of the rest of this talk. I'm going to focus on how cells build an embryo. And you might think, in the modern era, you might make the mistake of thinking along the lines of kind of a modern version of preformation. And that's this. You may have heard it said that DNA is a blueprint for building an organism. That's actually not right. DNA provides the information to produce macromolecules, large molecules, some of them carrying information, some of them ultimately making proteins that build an embryo. But the DNA doesn't provide the blueprint for this. Cells actually make themselves, and collections of cells that become embryos build themselves. And so DNA is part of the story, provides information. But it's not really a blueprint. Embryos form themselves in fundamental ways. So get that out of your mind right now. And then we'll move on and talk about how bodybuilding actually occurs in embryos. Maybe a better metaphor for bodybuilding in embryos is this. Has anyone done origami in their lives? I'm very bad at it. Here, this is your standard sort of crane origami. When you think about origami, it's a good metaphor for development. You're taking a flat piece of paper, you're folding it, you're making precise creases, and this leads to ever-increasing complexity. And in fact, if you've done origami, you know that the intermediate stages of building the structure that's going to arise from the origami doesn't actually look all that much like the finished product. But because of the process and the precision of the process, ultimately you get this amazing structure arising. Now, I was conferring with my colleagues over lunch today, and they said I must show the video that I'm about to show, so here it goes. It's a bit long, but it's kind of cute. Wait for it. Okay, now this guy's name is Rodolfo Rizzo, so he's Italian, so apologies to Gioacchino Rossini there, but you get the idea that, did you notice at the beginning, did anybody notice what happened before he started doing all of the construction? Anybody notice at the beginning? Yeah. How long did it take him to do the pre-creasing? 2 hours of pre-creasing before he did that. So origami is maybe not the best metaphor because actually embryos make their creases as they go. And in origami, you've got the hands of the person shaping the paper doing the shaping. Embryos shape themselves. But it's a pretty good metaphor of on-the-fly construction. That's what embryos do. Okay, what are some big ideas in developmental biology? We'll use some terminology, not much, but enough. One is differentiation. How do parts become different? Clearly that has to happen as embryos develop. But they do it in space, and that process is called pattern formation. Parts become different in a spatially organized way. And then there's the physical construction of the embryo. That's called morphogenesis, building the body. That comes from the Greek word morphe, which means form. And that's what we're going to focus on because that's what my lab studies, and I've been asked to talk a bit about the kind of science that I do. Now, we need morphogenesis along with differentiation. If you've ever built any Lego, my son had a lot of Lego, my older son John, and here's a pile of Lego. And you can see that there are differentiated parts there, but I think you can also see that there's not much of a structure there. So you need differentiation, but you have to assemble those differentiated parts in a very precise way for the body plan to be achieved. And that's what morphogenesis accomplishes. So what we want to consider tonight is what are at least a couple of ingredients that lead to successful morphogenesis. And it's at this point I want to tell you how I got hooked on this whole subject. I was studying the retinal pigment epithelium, the pigmented layer that lies behind the retina of the eye, and dissecting frog eyes in near dark conditions as a rotating graduate student at Berkeley. And I was hating being a PhD student. And I thought, well, what did I really like in college? Well, yeah, there was this course involving embryos. I don't really remember much of it, but maybe I'll see if there was a trainer in the biophysics program who does embryo work. And a guy named Ray Keller did embryo work. And I actually worked out a collaboration between Ray and a guy named Fred Wilt, also at Berkeley. And Fred worked on sea urchin embryos. And I took one look at developing sea urchin embryos through the microscope, and I was absolutely hooked. And so for me, that was the slip road to wonder as I began to appreciate the beauty and incredible complexity of embryonic development. So this is a sea urchin embryo here. You can see there's a lot going on, even in a simple organism. And I began to study something called gastrulation. We'll come back to this. It's a very complicated process. In the sea urchin embryo, this is the gut of this embryo, and it's doing stuff. You can see that there are cells crawling. Very complicated process. And I really have made it my life's work to investigate the processes of morphogenesis, these form-shaping movements. And it turns out when you look at this that it's a limited repertoire of movements. Cells can bend. They can wiggle between one another and make a longer structure in a process known as convergent extension. Sometimes there's a gap that gets filled in by cells migrating to the middle and making new connections by spreading. In other cases, cells leave a sheet of tissue, decide they're going to strike out on their own, and become invasive. And this has obvious implications for understanding invasive mechanisms in cancer as well, but it's a normally regulated process in embryos. And it turns out, if you look from embryos, from the simplest embryos, like little worm embryos that we happen to study on the right here, to more complicated embryos like this amphibian embryo on the left, and you look at places where cells are doing these kinds of things, the same cellular and molecular modules are in play from organism to organism. And so that the same machinery arises when cells have to pinch in a little worm embryo, or when they have to pinch in to form the central nervous system in a vertebrate. One of the things that cells have to do is crawl. And cells have a machinery for doing that. They assemble inside of themselves a cellular skeleton called the cytoskeleton, and one of the components of that is something called actin. And so here's a crawling cell in addition. You can see it's doing crazy stuff here. And it's extending what's called a protrusion. This allows the cell to migrate. So when cells in embryos migrate, they're assembling this very same sort of cellular skeleton. You can see how dynamic it is. Often, however, in embryos, cells are connected to one another and they behave as sheets. A fancy word for that is an epithelium. And in this case, you've got a sheet of tissue, it's got to buckle or it's got to do something fancy. And often that cellular skeleton will be assembled into a contractile apparatus, shown by the little red lines here. And if that happens locally, and the bottom edge of these cells tries to pinch off, just like the cinch on your backpack or something, you'll get a local bending in the sheet. And ultimately, this will lead to an impocketing. So in sheets of cells, you're dynamically assembling this cellular skeleton to allow movements, even when cells are connected. Now, if you think about this, it's great that cells can pinch off, but to transmit things to the entire sheet of tissue, all these cells have to stay connected. And so cells have to be connected by cellular adhesions to one another. I'm a textbook author of something called The World of the Cell. It's a sophomore level textbook. And we tend to design diagrams that are really boring like this, but you get the idea. It's very simple. It has that virtue. So here are some cells connected into a sheet. And you see these little green jelly bean shaped structures. Those are junctional connections that are filled with adhesion molecules, cellular glue that allows these cells to stay together. And if one of these cells decides to pinch off, then you can see it can pull on its mates and cause an impocketing. And if we go back to this diagram of all these basic repertoires of morphogenesis, all of them involve changes in adhesion in some fashion. In this bending case, these cells have to stay connected, so the pinching here is transmitted throughout the sheet. In this extension case, well, they've got to kind of dynamically make and break connections. Here, we're establishing a new connection where there wasn't one before. And down here, we're actually losing connections. We're dynamically breaking adhesions. So modulating cell stickiness is really important for embryos during morphogenesis. And in fact, cell adhesion is crucial for all kinds of processes. So as we've already talked about migration, how cells know their top from their bottom involves adhesion. When cells die, they often de-adhere from a sheet, and then they undergo a process called programmed cell death. It's important for organizing tissues, for signaling, and for general mechanical integrity. So cell adhesion is really important. What I want to do is to take a cell adhesion-centric view of the rest of my talk. And I'm going to focus on this. This is one of the core complexes that allows cells to stick together. It's called the cadherin complex. This is a molecule that's sticking out of a cell over here on the right, and the tip of another cadherin on an adjacent cell off the screen to the left is interacting with it. So cadherins from two cells interact through the parts of the molecule that's sticking outside the cell. And then the inside of the cadherin connects by some proteins called catenins to this guy over here, the actin cytoskeleton that I've already mentioned. And there's a protein called beta-catenin, which binds the cadherin, and then alpha-catenin binds the beta-catenin, the foot bone's connecting to the ankle bone here, and ultimately we're connecting to the cell's musculature, if you will, the actin cytoskeleton. So what I want to do is take a cadherin-centric tour of some key milestones in human development to convince you that this particular protein is super important. And so we're going to do that, and I'm going to focus on these first four stages. I was talking to Ruth earlier today. She said these sort of look like space aliens. You know, we all looked like this when we were developing our central nervous systems and our brain vesicles here. But I'm going to focus on early development, and in each of these phases of early development, cadherins play an important role. Then I'll talk about how my lab studies cadherins using little worm embryos, and then we'll move on to some wider implications of what we're talking about. Well, we all got, or well, most of us, I should say, can't say all of these days, but many of us got our start here. So oocytes are released from the ovary, and they're swept into this piece of plumbing here called the fallopian tube. And sperms swim up, and a lucky sperm encounters this oocyte and fertilizes it. And early development begins in the fallopian tube. The single-celled zygote divides several times, and then ultimately it becomes hollow with a group of cells at one end that we'll come back to. And slightly after this point, the embryo can implant in the mother's uterine lining. So this hollow structure is called a blastocyst. The division of cells in an early embryo is called cleavage. And so these cleavage divisions are occurring here, and ultimately the embryo hunkers down and attaches. So fertilization is obviously really crucial, and the sperm get through this protein coat and fertilize the egg. This triggers release of calcium ions inside the zygote, and this triggers all kinds of physiological changes and changes in the expression of genes in this early zygote. And this is what an early zygote looks like. These are the mother's contribution of genetic material and the father's coming together. Shortly after this, this embryo will begin to prepare for the first division, creating two daughter cells. After 2 such divisions, you have an embryo that looks like this. This is a 4-cell stage human embryo. This is the wall of the fallopian tube. And so this is a living embryo moving down the fallopian tube, eventually going to implant. And it turns out that going from a 1-celled zygote to the blastocyst involves these divisions, yes. But it turns out that cadherins are important for organizing the embryo during this very early period in its development. So here is a fertilized, or this is an oocyte. It hasn't been fertilized yet. After it's fertilized, it'll undergo several divisions. And actually, after 3 rounds of division, you have 8 cells, a little cluster here. And partway through this 8-cell stage, the embryo is going to become very compacted in a process called compaction. After additional divisions, some cells form at one end of the hollow embryo. That's called the inner cell mass. Those are very special cells. And then there are some blue cells around the outside, most of which will form the placenta later as the embryo develops. So you can see actual embryos down here, and you get the idea that it's not a lot of obvious difference here, but boy, by the time you get to a blastocyst, there's clear specialization of cells. And it turns out the cadherins are involved in this process of becoming tightly compacted, which in turn is required to get to this stage. So here's how this happens. So here's an uncompacted embryo, and here's a compacted embryo. You can see how much more tightly adherent this embryo is on the right. These are mouse embryos. Human embryos look very, very similar to this. And it turns out that this event requires cadherins. And you can show that by blocking cadherins. You can make antibodies against proteins, and then they bind the proteins and stop them from working. And here's what normal embryos look like. These are mouse embryos. But these mouse embryos have been treated with an antibody that prevents cadherins from functioning. And you can see that the cells look like a very loose cluster of grapes. It prevented compaction. And it turns out that that compaction process is important for helping cells in the very early human embryo to move towards one of 2 kinds of specializations. Before this time, it turns out that cells in the embryo can make anything. And in fact, monozygotic twinning is most often occurs prior to this compaction phase. And I just happen to like this beautiful photograph. And this involves a splitting apart of some of these early cells. But probably the best evidence that there are no specializations before compaction is this one. This is a great experiment from mice. You can take 3 mouse embryos and combine them. There's embryos 1, 2, and 3. You can see they're kind of loose. That's because compaction hasn't happened. But as soon as these embryos try to compact, they all compact together as a single embryo. So you get this big wad of cells, and then this thing will become a blastocyst, but with a very large number of cells at one end, these inner cell mass cells. Crazy. So you can combine embryos and they will form a unified single embryo. And if the 3 different embryos have different genetic constitutions, when you combine them, you can keep track of them. That's shown here. So you have mice that have 3 different fur colors. The chimeric mouse, the combination mouse, will have pockets of cells that have these 3 different fur colors, indicating the different genetic constitutions. This is all made possible by the expression of cadherin proteins, allowing these cells to compact. So cadherins are involved in compacting an embryo, and it turns out that at the next division, the cells in the interior of the mass of cells at the 16 and then 32 cell stage become very specialized. The ones in the middle become those special cells, the inner cell mass cells of the blastocyst. And the cells that are on the outside become something very different, the outer cells of the embryo. And this is way too complicated for us to discuss, except to say that the outside cells are connected to other cells on all sides, whereas the inner cells are connected by cadherins to all of their neighbors. And there's something that makes the cells different because of that condition. And this leads to the guys in the middle turning on genes that are normally expressed by these special inner cell mass cells. and the guys on the outside turn on a completely different set of genes appropriate to their position. So cadherins are important for establishing inside versus outside within the early embryo. And this happened to you when you were an early embryo. Well, eventually you get this. This is a blastocyst. And at one end of this is the inner cell mass, and that's what makes the baby, ultimately. So this material around here makes the placenta, which is left behind at birth. And these cells are very special. They are extremely developmentally flexible. You may know that stem cells can be derived from this group of cells. And whether that's something that we would want to do ethically or not is a great discussion to have. I'm actually not going to talk about ethics, hugely important topic. And these cells are at the heart of some of that ethical discussion. But they're very special for developmental biology because they can make so many things. And in fact, to make this blastocyst with these very special cells requires cadherin. So you can make a mutant mouse embryo that can't make its own cadherins and ask, how is it organized relative to a normal mouse embryo? Here's a normal mouse embryo over here, and these blue cells are inner cell mass cells. It's actually the nuclei of these cells. And there are some pink cells around the outside. Those are the ones that make the placenta later in development. Here's an embryo that can't make cadherins, and you can see it's got pink and green, kind of like this embryo, but I think you can see it's horribly disorganized, and the normal structure of the embryo has been lost. In fact, there are many fewer inner cell mass cells. And so blastocysts need cadherin. All right, so we talked about fertilization leading to cells dividing, proper division, and compaction requires cadherins. Eventually you get a blastocyst with these very special cells. That needs cadherins. And then shortly after the formation of the blastocyst, the embryo becomes layered and it undergoes a transformation known as gastrulation. And this is Lewis Wolpert. Some of you may know him from University College London. Lewis actually worked on sea urchin embryos at one point in his career, and he's an eminently quotable fellow, and he said this: "It's not birth, marriage, or death, but gastrulation, which is truly the most important time of life." Now, I've worked on gastrulation through much of my career, and so I'm sympathetic to this comment. I'm still mulling over whether I believe it. But gastrulation is clearly important for making an animal, because it's then that the body plan emerges. And my old PhD advisor, Ray Keller, has probably done the best work in a vertebrate. He's worked mostly in the frogs, Xanthus levis, to look at gastrulation. It is an incredibly complicated process by which cells on the surface are going to move into the interior, in this case behind the plane of the screen, and they're going to move asymmetrically to elongate the embryo along this dimension. And so let me just run this forward and we'll see how complicated this is. There's all kinds of things happening here. This thing is called the blastopore. Cells are streaming around that, sort of like Niagara Falls, and going into the inside. And you can see that the embryo is getting much longer. The head is going to form at the top and the tail at the bottom. Now at the beginning, this thing looks sort of like a beach ball, but by the end, it's clearly got a head and a tail. And that's what gastrulation does. It internalizes cells and transforms the embryo into a recognizable body plan. Now, it takes me a whole week to go through what's going on here with my upper division students in my developmental biology class. We don't have a week. So, happy to talk about it afterwards if you want. So there's very complicated things happening, and the details are slightly different in mammals, but mammals undergo gastrulation too. And so here's a cutaway view of a more advanced mammalian embryo. This is a human embryo diagrammatically. And you can see that it's connected to the uterine lining up here. That's what this red stuff is. So this is the placenta forming up here. The embryo is basically a double-layered flat disc with two balloons on either end. The top balloon is called the amnion, the amniotic fluid. That's when a woman is about to give birth and we say her water breaks. It's that water balloon that breaks. And then there's another balloon down below called the yolk sac underneath. So, but if you kind of strip away this mentally and you just look at this disc, it's doing movements similar to that frog embryo I just showed you. So here is this double-layered disc, And if we look down on the surface, there are cells that actually dip down through a central depression called the primitive streak and move to the inside to create a middle layer that was not there before. The outer layer is called the ectoderm. That forms things like your central nervous system and your epidermis. The lower yellow layer here is called the endoderm. It forms your guts and a lot of your internal organs. And this middle layer, the mesoderm, forms as cells stream through this streak of material. These movements are incredibly complicated, and no one really understands how they work in amniotes. That's things like reptiles, birds, and mammals. But Case Virus Lab at the University of Dundee has done some really nice work in chicken embryos, which geometrically look very similar to human embryos. So I just like this movie. It's a gratuitous movie, but gratuitous movies are good. And this is tracking some fluorescent cells that move into the inside and then stream up towards the head end of the embryo. And you can see these comet trails that track the movements of these cells. Just give you the impression this is an incredibly complex process. And you can imagine that there's changes in adhesion that are involved. Clearly cells are migrating, and they have to be guided to do so. Complicated choreography. Amazing. Well, eventually, after gastrulation is over, we all look like a burrito. Maybe, I guess apparently some people don't eat Mexican food a lot here in the UK, so maybe a falafel wrapped in pita or something like that. And the head end of the embryo is over here, tail is here, and you can see that the embryo has wrapped up on its outer surface, and this is the formation of its central nervous system during the process of neurulation. Neurulation is a great one of these folding movements of morphogenesis. It involves a flat plate of tissue, this ectoderm on the outside of the embryo, and it's going to bend in the middle. So it's going to fold. You can see that the aqua regions are going to approach one another, and actually they meet, and those cells detach. They form a very important group of cells called the neural crest. Without the neural crest, you wouldn't have much of your face, a lot of structures in your body. They migrate all over the embryo after they meet at the middle. And then the blue on the extreme edges here, that's epidermis, and that has to seal over and fuse to encase this tube, which is now formed in a layer of skin over top. So here's neurulation in a couple of salamander embryos, and you get the idea. This is marvelous. The heads are up here, tails here, and you can see that these are self-rolling burritos, in fact. Whoops. And here we go. And you get the idea, right? That they are, these folds are meeting, and then ultimately we have an embryo that from head to tail has a central nervous system. So you see how complicated this process is. And yet it's really pretty simple morphogenetically. We have a flat sheet, it's folding, and then the folds are meeting over top. Now the same thing happens in higher vertebrates. Here's a chick embryo, and it's hard to see. The optics are really tough trying to make movies by opening up the egg shell of a chick and then doing time-lapse filming. But you can do it. So here's the movie. And the head end is over here, tail is here. And you can see that the brain is already forming before the back end is finished neurulating. There's a big temporal difference in how that works in birds and mammals. But you can see here, see how these folds are going to meet in the middle by the end of that movie? So you get the idea. And humans do the same thing. So humans are sort of a bumpy burrito, I suppose, and these bumps are called somites. They form blocks of tissue that form many different structures along the body. But the same thing happens. The neural tube forms, and it's originally open at either end. Now, if you don't close both of these ends, disaster can result. Particularly if you don't close the posterior end, you have a common birth defect called spina bifida. So that fusion event is very, very important for ultimate health of the spinal cord. And you can see there's a little protuberance. So that's a part of the spinal cord that's bubbled out because the skin didn't seal over properly. All right. Now, in my opinion, we've had some nice movies, right? Would you agree so far? These next two, I think, are the coolest I'm going to show. And these are mouse embryos doing neurulation, labeled with fluorescent molecules so you can see them. This is from Lee Neiswander's lab at the University of Colorado, Denver. You see how these cells are zipping up? They're going to meet. So these cells are approaching one another. Here are the edges of the neural folds. You see how much they have to move to get to the midline. The anterior neuropore, the open end near the head, is enclosed by the end of this movie. But there's all kinds of crazy stuff happening here as these cells are interacting with one another. If you look a little later, here's a mouse embryo in which neurulation is almost over, and this is the only opening that's left. You see how it closes off. So you can see you've got to make new connections if you're going to complete neurulation. And it turns out that cadherins are involved in neurulation as well, not surprisingly. In fact, if you look at the neural tube, that part that forms the tube part of the burrito, and you look at the future epidermis out on the edges that seals over that tube, they express different cadherin proteins, different flavors of cadherin. So the neural tube expresses something called N-cadherin, N for neural, and the epidermis expresses something called E-cadherin, for epidermal. And expressing the one here and the other here turns out to be important for keeping these two tissues separate so that you can complete neurulation. And if you force an embryo to express the wrong kind of cadherin in cells where it shouldn't, disaster results. And this is a frog embryo. And the blue cells here have too much N-cadherin when they shouldn't have it. And you can see that the neural tube never really closes, and disaster results. So cadherins are important in all of these different steps of embryogenesis. And that's where my research comes in. So, I'm sorry, this is from a rival institution from a couple of years ago, you'll see why I picked this picture in a moment. But we actually work on the cadherin complex. But we use a non-vertebrate organism to do that, a nematode worm called C. elegans. So adult C. elegans are about 1 millimeter long. Embryos developing inside of the self-fertilizing worm are these little bean-shaped affairs here in the middle. Now they're gray. They don't have very many cells, about 1,000. Every embryo develops into an adult in exactly the same way. So if you perturb something, you know that you've perturbed it. They're transparent, so you can watch them develop as living embryos. And you can do powerful genetics. Now, we studied the embryos that have about 500 cells. Very simple compared to the mouse embryos that you were looking at earlier. But they do a lot of things that more complicated embryos do. You can see that cells are migrating. They're going to meet on the midline. The whole embryo is turning on its side. This is the head, and this is the tail. You can see it starts to twitch because muscles develop, and then it begins to elongate. So you can see that it's sufficiently complicated that it has enough richness to keep us occupied for a career, but sufficiently simple that we can understand what we're doing and understand what happens when we perturb particular molecules. This is what developmental biologists call a model organism. The basic idea here is that you can learn a lot about a Maserati by studying a Toyota. If we're the Maserati, these little worms are the Toyota, or the appropriate UK economy car of your choice. And in particular, we can do molecular and genetic analysis that are simply not possible in vertebrates, nor would they be desirable in humans. And to illustrate this idea, this is my son John and his wife Mary. So this is my older son John. Christopher's my younger one. And well, they got married in 2011. This is at the wedding reception. This is Dad's car on the left. Actually is a Toyota Corolla, economy car. This is my son's car on the right. Custom license plate says Hardin. This is a Maserati. He now owns apparently a Jaguar that is similar to the one, it's not bulletproof like the Queen's, but apparently similar to the Queen's. That's all I know, don't ask me. But the idea here is that if you look at the engine of this Toyota, the operating principles are very similar to the Maserati. Yes, it can't go from 0 to 60 quite as quickly, but many of the basic principles are the same, and that's what these worms get us. And in fact, worms have a great cadherin complex, and the proteins have names. The cadherin is called hammer 1. The catenins are called hump 2 and hump 1. Hence my little traffic sign. Make sure a few slides back. And cadherins are expressed and deployed in ways that are very similar to what I showed you for human embryos or mammalian embryos like the mouse. The green here is a beautiful picture from Jeremy Nansen's lab at NYU in New York City. Shows cadherins between cells in an early embryo. And we've done some work to show the cadherins are deployed and work in the same way as we've seen in other organisms at this very early stage of worm development. Later, when the embryo is elongating, it has really beautiful junctions connecting all of the cells. And so this is from my lab, and you can see that the green is the cadherin complex connecting cells in the epidermis, the skin of these embryos. In fact, that's what we study most of the time. Worms are shaped like a bean, and the epidermis consists of about 80 cells, which covers over this bean and then transforms its shape. And in particular, the pink cells here migrate to pull the epidermis around the embryo to cover over the nervous system. So this is the neurulation equivalent in a worm embryo. And the epidermal sealing that occurs here is very similar to what has to happen during neurulation, for example, and has lots of implications for understanding how wounds close and things like that. So here's a movie showing this. We can analyze this at very high resolution. So here you see some cells migrating to the middle during ventral enclosure, and they're meeting and establishing new junctional connections along the ventral midline, the belly part of the embryo. And this requires the cadherin complex. So here's an embryo using one kind of microscope on the left and looking for a protein that's part of the cadherin complex on the right. And you can see as cells migrate and meet at the middle, right in here, you're going to see a line of cells right now, and they're making new connections which we can watch form on the fly using something called a spinning disc confocal microscope. Remarkable. And similar to what we've seen with other more complicated embryos. Now, these genes get their names because of what happens to embryos that can't make these proteins in worms. So when embryos can't make cadherin, they try to do ventral enclosure, but they can't seal. And so when they try to elongate into a worm shape, they explode. And that's what we call the hammerhead phenotype. Cara apparently spent a lot of time in the Maldives. I don't know if there are hammerheads there, or Cara, no. Now, if you can't make catenins, then you complete ventral enclosure, but when you try to elongate, your actin pulls loose from your cell surface because it's not connected. And you develop these dorsal humps, which we call the humpback phenotype. So these proteins are essential for these little worms. And here's some movies. So here's a hammer 1 mutant embryo over here on the left. This is a hump 2, this is beta-catenin mutant on the right. Watch this guy. Unfortunate things about to happen. Here we go. So he's spewing out his innards. And yeah, he's not going to make it. The guy on the right is not able to elongate, and he develops these dorsal folds up on his dorsal surface, his backside. So this is easy to see. Whenever we see it, we know that it's happened. And what we've learned is that, in fact, as cells make these new connections, by watching them very carefully, is that they load cadherins into these crawling parts, so that when these crawling parts meet to seal the sheet, they rapidly deploy them to rapidly make a connection. The other thing we've used these worms for is to understand the wider network of proteins that embryos need alongside cadherins. And it turns out that almost all of the things we discovered are conserved from worms up to humans. So humans have very similar proteins, presumably using similar rules. Now here's a lesson in genetics, the only real didactic lesson for today. You ready? You can do it. Let's say I have a junction, shown by this Picasso art book, Picasso at 90. And let's say that it's connected via catenins, in particular alpha-catenin here, shown by this particular man-made object. But suppose there are other supports that assist with supporting the junction. If we remove those other supports, if we have a fully functional alpha-catenin, then we can connect to the actin cytoskeleton, transmit forces, and do morphogenesis. But now suppose we had a less than fully functional alpha-catenin. Now if we remove these supports, disaster. And it turns out that we can look for things that behave in this way. So now I'm going to ask you to do some morphogenesis detective work and see if you can see some differences. So here's a normal embryo on the left, elongating. And the embryo on the right has a partially non-functional version of alpha-catenin. You can see, it's trying. And actually, a lot of these guys will stay alive and form really nasty-looking hapless adults, but they're fertile, and you can keep them going. It turns out what causes this is a particular building block of this protein, an amino acid, that humans have in exactly the same spot. That's a story for another time. So here's the embryo that was on the right, now I've moved it to the left. And now we're going to get rid of another component of the cadherin complex that's not essential normally, called JAK1. All I want you to be able to see is that they're different. This guy's developing those dorsal humps. That's the humpback phenotype. So he looks a lot worse than this guy on the left. So it turns out you can do this for any gene. You can look for every single gene in the worm's genome and its genetic constitution, that when you get rid of the function of that gene and the products that it provides the information to make, does this. And worms have a really nice trick that allows you to do that. So in the lab, worms eat bacteria. They live on their food, eat it for breakfast, lunch, and dinner. Kind of a nice life. You can feed worms bacteria that are making something called double-stranded RNA, that all you need to know is, if the worms eat bacteria that are making this, they transfer these molecules to their gonads, so that when they make their offspring, those offspring lack the function of the thing you put in. This is called feeding RNA-mediated interference, or feeding RNA out. You can remove genes by feeding, okay? Very cool. So this is what we did. So you can take these worms they don't do adhesion as well as normal worms, and you can give them bacteria to eat that cause their offspring to lack the function of particular proteins. And here's what we're looking for. We are looking for a double whammy here. When you've got this first problem with your adhesion, and now we take out some other molecule at the same time, all the offspring die in the way that I showed you in the movie. And this is tedious but straightforward. So we did this. You can go through, we went through 16,500 clones, most of the genes in this little worm's genome, and we got about 60 things. And those 60 things, we've characterized some of them, but it turns out that the vast majority of these have human counterparts. So this validates the whole model organism idea. Now, the last thing I'll tell you about that we've been doing is something that you can do in worms that's really great. You can make new worms, and then you can put in bits of DNA that provide the information to make parts of proteins. And you can ask, which parts of the protein do we need, and which don't we need? And so that's something that's called structure-function analysis. And all you need to know is we can do that. And the other thing that you can do is we've been able to purify the proteins of the cadherin complex and determine their structure using something called X-ray crystallography. So we can get down to incredible detail and ask, well, how similar are they to human proteins, and then what happens when we mess up particular parts of these proteins. And that's something I call the angstroms to embryos approach. Angstroms are really short distance. And this allows us then to understand how the great details of this complex are required for functions of more complicated organisms. And here's one example of doing that. So these these cylinders is beta-catenin and has a particular structure. And you see there's a rainbow-colored squiggle. That's a piece of cadherin, the worm cadherin hammer 1. You can see the squiggle fits in the nooks and crannies of these cylinders. Colleagues who are X-ray crystallographers, if they heard what I just said, they'd be pulling their hair out, but that's all we need to know. Now, if you look over here on the right, see that gray squiggle? That's the structure of the human E-cadherin, epidermal cadherin, as it interacts with human beta-catenin. And the gray and the rainbow-colored squiggles lie right on top of one another. They are identical down to the level of individual atoms in the structure. That is amazing and very gratifying. And what we can do then is, based on this, we can cause mutations. We can disrupt particular positions along this structure and ask how well do those proteins work. And it turns out this one right here, if you mess with that one at all, the cadherin can no longer attach to beta-catenin. So this is like a little linchpin that allows cadherin to interact with beta-catenin. All right, that's a lot of detail. All it says is that worms do things that are really similar to humans. And in fact, there's a reason for that. That's because both worms and humans reflect a deep evolutionary history. You can look through the animal world and find cadherins in all multicellular animals. And you can even find cadherins, they don't have all of the bells and whistles on their inner portion compared to multicellular organisms, but they're even found in cousins of animals called choanoflagellates. So these are really interesting creatures. They look like a part of a sponge. It's the part of a sponge that sweeps food through the sponge and it siphons, but they're just that part alone, so they've got this flagellum. And the red part is the choanoflagellate's cadherin. And it's thought by Nicole King's group at Berkeley that this is important for them capturing bacteria, which is what they eat. But some of these choanoflagellates can be induced to aggregate. So they're beginning to develop the possibility of making multicellular structures. Now they diverge from animals between 750 and 1,000 million years ago. And that is a long time. Here are sponges, and they have what looks like a pretty respectable cadherin. Next up are these things, really crazy, weird animals called placozoans, and they have the first real tissues that anyone's been able to see. So cadherins are found in all of these organisms, and you can trace a family history of cadherins, and it matches very, very well the family history inferred from other kinds of criteria that have established evolutionary relationships between all of these organisms. So the cadherins that we have reflect this deep evolutionary history that goes back to the dawn of multicellularity. Well, that's all I want to say about cadherins. And what I'd like to do for the last few minutes that we have is to focus on the wider context. So here's my office at work. You can see my office is no great shakes. It's early 1960s kind of penitentiary architecture, which was en vogue at the time. But this is the window, which looks out on a beautiful parking lot, by the way. Now there are three objects in here that I want you to look at. There's one on the right, one on the left, and this little one in the middle. And art imitates life, I suppose. The poster on the right is a poster of some hand drawings by a guy named Theodore Boveri of early nematode development, worm development, in the same grouping of worms that C. elegans, the model organism I use, is found. So that's the science. I'm actually on the editorial board of that particular journal too, which is another reason why it's there. Does anyone recognize this? This from the Lindisfarne Gospels. And so I got this. This was before the British Museum and the British Library were separated physically. Tragic. Anyway, this reflects the fact that I'm a Christian and that my study of the natural world is part of my study of God's world. Now in the middle was this is a little thank you note from somebody at the BioLogos Foundation. I happen to be the chairman of the board there, and this is an organization in the U.S. that has some overlap of mission with the Faraday Institute in that it's trying to help people to understand that there can be a harmony between good science and deep faith. And so this is my approach to thinking about my life as a scientist and as a Christian. And for me, doing science, every time I peer down a microscope, is an exercise in worship. I can affirm with the psalmist that I am fearfully, wonderfully made, and my soul knows it very well. And that is a gift. To be able to practice science as an exercise in art appreciation, an appreciation for the marvelous created order that exists because of God's activity. So that's how I view my own career. But it's true that there are some issues. So here's another eminently quotable fellow who liked his beer, Martin Luther. And Martin Luther said this. Well, okay, it's hard to know whether he said everything that's in the table talk, but it sure sounds like Martin Luther. The reproduction of mankind is a great marvel and mystery. Had God consulted me in the matter, I should have advised him to continue the generation of the species by fashioning them of clay. As I should have counseled him also to let the sun remain always suspended over the earth, like a great lamp, maintaining perpetual light and heat. Well, fortunately, Brother Martin was not in charge of celestial objects, and he's obviously not in charge of embryonic development. We know it's incredibly complicated, so we have to come to terms with what we've learned and what it tells us about how God acts in the process of generating animal body plans. And one of the things that I guess is possible is that you could think that maybe what we know leads to further alienation. Science has told us a lot, and in the face of science, we seem sometimes fairly insignificant. And starting as a one-celled zygote here, knowing what we now know, adds perhaps to that sense of insignificance. We're actually suspended between two extremes, the very big, the very old, and the very fast and very small. Just 3 examples. There are 300 sextillion stars in the universe. Well, that number keeps changing, but it's close enough. That's a really big number. Very big number. 6 times 10 to the 23rd. Or 30 times 10 to the 23rd. That is a really big number. Here is the age of the Earth at the time that life first appeared, approximately 3.5 billion years ago. Bob will tell you that that's a really long time, and he knows a lot about that. And then here we are. We all began like this, 100 micrometers in diameter, and yet we become 27 to 50 trillion cells. So we start in a very small, insignificant way. And there was a really smart guy who understood this. He was way ahead of his time, Blaise Pascal. And he said it this way, look, let's take our compass. We're something, and we're not everything. Pascal clearly did not know about the blogosphere. We'll let that pass. The nature of our existence hides from us the knowledge of first beginnings, which are born of the nothing, and the littleness of our being conceals from us the sight of the infinite. We know that we're small, and we know that we come from things that are small. So this could perhaps lead us to think about ourselves as insignificant. And there's a sister problem here, and that's the fundamental thing that biologists do. And that's to think reductionistically. Lots of other scientists do this as well. We poke apart cells, we pry into the genomes of these cells, we see what happens. We develop mechanisms with flow charts and arrows and molecular maps. Another guy also way ahead of his time is the metaphysical poet John Donne. He said it this way: new philosophy calls all in doubt. The element of fire is quite put out. The sun is lost in the earth, and no man's wit can well direct him where to look for it. 'Tis all in peace, is all coherence gone, all just supply and all relation. That's the danger of reduction, thinking we're nothing but a collection of cells. Is that what we're left with from this analysis? Well, I would argue not. And this is where religion provides an important remedy to what we've been talking about. Former rabbi of the UK, Jonathan Sacks, in a really fine book called The Great Partnership, on page 2 of that book, said this: science takes things apart to see how they work. Religion puts things together to see what they mean. And that might be oversimplification, but it gets at something. Science is good at this first thing. It really can't address the second thing. And that's where my Christian faith is hugely important. Simone Weil said it this way: if I light an electric torch at night out of doors, I don't judge its power by looking at the bulb, but by seeing how many objects it lights up. The brightness of a source of light is appreciated by the illumination it projects upon non-luminous objects. The value of a religious, or more generally, a spiritual way of life is appreciated by the amount of illumination thrown upon the things of this world. Religion provides the meaning that science never can. Significance is a judgment about meaning. Science can't provide that judgment, but religious perspectives can. And in particular, my perspective is that I am a practicing Christian. And another piece of poetry from the Hebrew Scriptures puts this well. When I look at your heavens, he talking to God, the work of your fingers, the moon and the stars you've established, what are human beings that you're mindful of them? Mortals that you care for them. This idea of looking out at the world and feeling small is not new. But the psalmist was onto something, was onto a secret about the source of significance. A way of seeing ourselves that the scriptures can provide. And in fact, the scriptures tell us something else. I would say one of the things that nature does provide us as we study it is a sense that we are fragile and small. That ought to lead to a character quality and an epistemic quality, humility. Now, I have a lot of scientist friends, my colleagues. I would say as a group, we're not strong on the humility thing. Let's just be honest about that. And yet as a Christian, I recognize that the universe is vastly greater than myself. That I'm part of something enormous and deeply meaningful, something that will last far longer than my own lifetime. Andrew Murray, pastor from South Africa in the 19th century, said it this way: there are three great motives that urge us to humility. It becomes me as a creature, as a sinner, and as a saint. Studying embryos gives me a sense of humility about myself as a creature. You need only ask my wife about that second part, that sinner part. She'll tell you all about that. But as a Christian, I see that from my humble beginnings as something 100 micrometers in diameter, God has a plan for me that is vast, sprawling, and wonderful. Well, I mentioned my wife. Here she is. This is Susie. And she's my practical theologian. In particular, there's one other thing we have to talk about. When a baby is born, we often say life is a miracle. In fact, there's a great video called The Miracle of Life. And you may have heard that phrase. Well, what about what we learned? Well, I would argue that the amazing processes that underlie embryonic development and indeed all of the deep evolutionary history of life on this planet are a magnificent vision of how God operates. Now, Bob mentioned that's Darwin's birthday, and we have Darwin Day celebrations at University of Wisconsin, and my department, the Department of Zoology, is engaged in. And theologians in the UK were at the forefront, I think, of grappling with what Darwin was saying, what it meant. One of them was this fellow, Charles Kingsley, and he thought a lot about what's called natural theology, how God works in the world. He said it this way: are we to reverence him less or more if we hear that his might is greater, his wisdom deeper than we ever dreamed? We knew of old that God was so wise that he could make all things. But behold, he's so much wiser than even that he can make all things make themselves. Now, he was talking about evolutionary diversification of life on this planet, but the same is true for embryos who make themselves. And I see this as a grand way of thinking about how God works in the world. All right, finally, Christianity has a unique idea, and that is that the second person of the Trinity somehow entered into human form and lived among us. And he did that as an embryo. And so for me, thinking about embryonic development fills me with an ultimate sense of wonder and awe at how this is all played out. Lucy Shaw said it this way: after the bright beam of Annunciation fused heaven with dark earth, his searing, sharply focused light went out for a while, eclipsed in amniotic gloom. His cool immensity of splendor, his universal grace, small folded in a warm, dim female space. Understanding the complexities of embryonic development and understanding that God knows intimately about that process fills me with a sense of awe and a sense of profound wonder and worship as I study these processes. Thanks very much for listening. I hope this has been helpful to you, and I'm happy to answer questions. And I'm also happy to entertain more questions out in the lobby during our social time together. Thank you very much.
Thanks. Well, we'll take a few minutes for questions now, before lots of hands already before we start. Somebody's got a microphone. You're going to get it. Oh,
I think they want you to wait for the microphone so that we're recording this, that they can hear the questions.
In the middle.
Let's go up there, up towards the back.
Yeah. First hand up. Third row forward. Quick, first in front. Yeah.
So I'd like to pick up on there were
Can you use the mic?
Excuse me. So with regard to 2 points, you had said earlier in the talk that the DNA of the embryo is not a blueprint. Then we had Westminster Simmons quote toward the end. There was God making things make themselves, and you had all this discussion about the physical processes of what's going on in embryonic development. And my question is, to what extent does the embryo bootstrap itself from the cell, and to what extent is the maternal environment acting as a chaperone beyond the obvious things of respiration and temperature control and that type of thing? How much can the embryonic cell do by itself if it's just given a reasonable environment, and how much does it rely on more detailed maternal processes?
So let me clarify one aspect of that question. The embryonic cell, you mean the one-celled zygote?
Whatever point you want to answer, that's interesting. I'm just, anything going along the lines is...
Yeah, so there's a real chicken or egg problem here. And that is that I didn't talk about a hugely important process for early embryonic development, and that is that mothers make oocytes or eggs. And they put all kinds of interesting molecules into those eggs. And without the mother's production of a properly functioning oocyte, the fertilized oocyte can't develop. So there are many gene products, which are called maternal effect gene products, that are put into the oocyte. So that's what makes it tricky to think about how evolution acts on early development, actually, because it's clear that the complex of oogenesis plus what you get after fertilization all feeds in to what you can select for in terms of a body plan. This is not to say that genes are not the crucial thing. I'm more of a traditionalist in terms of evolution than some people who talk about the new extended synthesis, I suppose. But which side you fall on in terms of that debate, no one is questioning that you've got a complex of things going on. One of the things we're discovering, of course, is that the DNA that comes into the oocyte, or the DNA brought in from the father's sperm, are actually chemically conditioned in a process known as epigenetic programming. It's a use of epigenesis different from how Aristotle used it. But it means that it's not just the sequence of DNA, but the chemical status of the DNA and associated proteins that it's wrapped around, called histones, that helps to determine the availability of particular pieces of chromosomes to be expressed. And that reflects the prior history of the mother and the father. So there is some bootstrapping that goes on, to be sure, but you can show very, very convincingly that the embryo has to transcribe its own genes, and that happens in humans and other mammals at the one-cell stage. In the little worms I study, happens a bit later. So, yeah, I think, so there's a lot going on there. And I'm sorry, I've lost the train of thought if there was another part to your question. Good? Great.
Well, let's go to the back. Yeah, thanks.
Thanks for that detailed and music talk. Could you please help me respond to this in a very simple way I could understand, with due respect? Were you stating that the genes carry parts of the information for constructing the cell, or that this information is distributed between the genes and part of the cell?
Well, the information that can be transmitted from generation to generation is provided in the DNA. But I did say, and that's why I made the point earlier about that DNA is not a blueprint. You can't go from there are intervening steps that have to occur. The DNA provides the information to make proteins, but then once those proteins are made, they interact with one another in very complicated ways and localize to particular spots in an egg, for example, or in the cells of a developing embryo. So it's not always straight. You can easily determine what happens when you mess with a particular gene. So if you look at the end product, you can say, I messed with that gene, oh wait, now I don't get any skin, let's say. You can do that kind of an experiment. But the intervening steps involve interactions between cells, so it's not totally straightforward how one goes from the sequence of a DNA to how that information is read out and how the assembly process works to generate the form of the embryo. So DNA is here to stay. It's crucial, and it provides the information for the building blocks in the embryo, but once they're made, they have to assemble, and that doesn't involve the DNA directly.
Thanks very much. Based on what you know in biology, would you care to estimate the chromosome count in both the gametes and the autosomes of Jesus Christ?
I would not care to do that. Actually there was discussion, so St. Thomas Aquinas had a whole theory about at what stage of development as a fetus was Jesus placed into Mary. So there's a discussion there that we had. I'm not going to.
Surely you're more qualified. More information to be taken about.
As a Christian, I'm comfortable with God intervening in certain key events in the history of life on this planet. That is one of them that I'm comfortable with. Yeah. But there are people who want to theorize about how it could all happen, involving parthenogenesis, but you have to get a Y chromosome in there because Jesus was male, and how do you do that? And I prefer not to speculate.
Hello, I have two questions. The first question is about developmental biology. I learned long ago that when a bird's egg hatches into a chick, the distribution of protein within an egg affects the expression of genes. Is it caused also by DNA? And the second question is, up to what stage, or what developmental stage of an embryo that you would consider it live, that you would stop experiments using those embryos?
Okay. So it is true, this is getting back to the question earlier about bootstrapping from an egg to an embryo. There are certain molecules that must be placed precisely in many different animals' eggs in order for the subsequent animal embryo to develop properly. Absolutely clear. Absolutely not clear in mammals whether that works. So we don't know. In birds, there's some process that happens normally as the developing egg, before it gets its shell on, is going through the oviduct, the plumbing, and the whole thing twists and torques, and that twisting in the oviduct determines where the posterior, the back end of the bird embryo is going to be. Nobody really understands how that one works either, but clearly there's something going on with the ovocyte before it ever gets fertilized by the rooster, right? So, and that's good evidence that something must be happening there. Okay, so the second question, I've chosen to work on an invertebrate model organism that is not actually considered an animal by my university. And I did that because it doesn't have a backbone. So, you know, our university has regulations that mostly involve a development of central nervous system function, at which time the regulatory processes kick in. And this starts with fish, and then goes all the way up to mice. Humans have there are a few people at my university who work on human embryos, mainly stem cell people, and they obviously would never allow an in vitro fertilized embryo to get to that stage. So, you know, this is one of the interesting issues that we simply didn't have time to cover, is what is an organism defined by its set of functions, or is an organism defined by its organismal capacities? It is not, the answer to that is not obvious. So for humans, is there a sine qua non of human embryonic development, at which point you say, that is a fully protectable human individual? Or do you say, all human organisms are worthy of protection to the extent that we can, because they have the capacities to develop all of these human functions associated with more mature humans? That's a huge ethical contention, and I think people don't agree on that. But that's a key set of questions that one must try to answer if you're going to develop a robust answer to the question you asked.
I think that's a very weighty point on which to stop, because there are drinks waiting next door, and Jeff will be very happy, as he said, to carry on chatting over drinks. So let's thank him again, and I know that Hilary wants to say a word, but we'll thank him again, and Hilary's going to explain.